Luciano, I'm looking forward to hearing from you, and the floor is all yours. Thank you very much.
And thanks a lot, Francis, and I would like also to thank Motif for organizing this meeting and to ask me to share this very interesting session. Hope you can see the slide, and also can you see my mouse?
Yes, Luciano.
Yeah, perfect. Okay. So today I would like to talk with you about the role of Polycomb and the opposite complex, which is the MLL Trithorax complex in stem cell and cancer. So Polycomb comes in two flavors, the PRC1 complex and the PRC2 complex. The key component are, the core component are listed here. And Polycomb was identified in Drosophila many years ago, and as the group of protein which counteract the activity of trithorax in regulating the expression of the OX genes. So trithorax, which you will see later in mammalian cells, is called MLL complexes. It's a multi-protein complex which lead to gene activation, while Polycomb, which is the opposing function of trithorax, leads to gene repression to a variety of mechanisms, which I don't have time to discuss here, but if you want, we can discuss during the question session. So as I said, Polycomb comes in two flavors, the PRC1 complex and PRC2 complex, which can again then subdivided in the PRC1 and the canonical PRC1 complex and non-canonical PRC1 complex, while the PRC2 complex comes in two different compositions, which are the PRC2.1 and the PRC2.2. So this is like a little summary slide, then I will go more in details on the mechanistic implication of this, but just remember PRC1 complex as two, the canonical, non-canonical, and PRC2 as the PRC2.1 and PRC2.2 variations. Okay, mechanistically, the way in which Polycomb repressed transcription is, according to the classical model, is that the PRC2 complex, which again, some of the core subunits are listed here, leads to deposition of trimethyl mark on lysine 27 of histone H3. So this trimethyl group on lysine 27 is an repressor signals which lead to chromatin compaction, again, by different mechanisms. But also what it does, it is creating a docking site for the other Polycomb complex, the PRC1 complex. So as you can see here, the PRC2 complex deposits the mark, which is then read by the PRC1 complex, and the combination of PRC1 and PRC2 are chromatin leads to chromatin compaction, repulsion of polymerase machinery, and other things, which at the end leads to gene silencing, what leads to maintain the repressive state of a given genes. More recently, the work of several labs, including that of Robert Close, highlighted is a PRC2, PRC1 axis actually can also be reverted, where the non-canonical PRC1 complex lead to the position of H2A to a monoubiquitin group of histone H2A, which then recognized by JARI2, which is one of the components of the PRC2 complex. So as you can see here, the order of recruitment is the inversion, is inverted one. So first PRC1 complex and then the PRC2 complex. Okay, so as I said, Polycomb repressed transcription, and this is done by deposition of several histone modification. I listed here K27 trimethylation, H2A monoubiquitination. And these usually the Polycomb repressed genes, which are implicated in cell differentiation and lineage specific genes, and those genes are maintained repressed, as I said, by the presence of polycombs. Now, this repressive mark can be erased, and the complex which erases the repressive marks actually is the anti-Polycomb complex, which is called Trithorax in Drosophila and MLL complex in mammalian cells. Together with Ramin Shikatar, we purify one of these variation of Trithorax complex, which actually is very interesting because contain UTX, which is enzymatic enzyme, which is capable of erasing this histone modification, which is deposited by Polycomb. But then you also have the MLL subunit, which is a cytodomain enzyme, which can lead to deposition of an active mark, which is K4 methylation, as indicated here. So as you can see here, this complex is capable of erasing repressive mark and also leads to activation of gene. That's why how Trithorax leads to gene activations. Now, if we look in the stem cell, stem cells, as you know, are derived from the inner cell mass, and they are capable of undergo to differentiation in many cell types. But the way in which are culturing the cells, these cells can proliferate forever and actually do not undergo anti-differentiation. So the reason for that is because the stem cell genes are active, and those include the pluripotency network, cell proliferating genes, and so forth. And those genes are bound by MLL complex, Trithorax MLL complex, those genes, as I said, are on. While lineage-specific genes are maintained off by the occupancy of Polycomb complexes, both PRC1 and PRC2. Now, both in vivo and in vitro, when we trigger differentiation, what happens is that those genes which contain Trithorax and were active upon induction of the differentiation stimuli, Polycomb relocate to these genes and leads to the repression of these genes. And Trithorax now is capable of activating a specific set of genes, which in this case may, for example, may lead to hematopoietic differentiation. Now, if we stimulate differentiation toward, for example, epidermis stem cells, a different set of genes become free of Polycomb and become now target of Trithorax, and those genes become activated, while pluripotency genes become downregulated. So this is the way in which cells lose pluripotency and gain specificity toward a given lineage. Now, what I've been telling you is that Polycomb Trithoraxes have opposing function. Polycomb repress transcription by depositing this modification, and Trithorax activate transcription by depositing this modification. So the reason why we work on Polycomb and Trithorax is not only because it's very important for animal differentiation, but also, as also was shown before by Paola, this is a different study, but again with the same conclusion, that if you look at a large number of different types of tumors, among the top proteins which are modified, this large number of tumors, you can see Trithorax complex, MLL 2, 3, and 4, as well as Polycomb proteins. So they're often mutated in cancer, but not only in cancer, also in many other diseases. Now, if we focus on the PRC1 complex, then here, this is the canonical one, the core subunit are listed here. We know that there are, for each of these protein, there are many paralogs protein. For example, ring 1 can come in two forms, ring 1A or ring 1B. The same is true for CBX protein, can be CBX 2, 4, 6, 7, and 8, and so forth. So in order to assemble the PRC1 complex, you can consider all the possible combinatory assemblies. So for example, you can have PhfI9, PhS1 together with ring 1A and CBX2, or the same CBX4, CBX6, and so forth. So if you calculate all possible variation, although we know that not all possible variation are actually can coexist, we know that the overall number could be up to 200 variations. Are these 200 variations really, do they have different function? Do they bind to different set of genes? And how the shift between these variations is regulated? Why sometimes you have CBX7, sometimes you have CBX2, which is the PRC, canonical PRC1 complex. So we have addressed some of these questions. You can see some of the recent publications from the lab in try to addressing this complex, this question. And for example, what we learn is that in pluripotent stem cell, the composition of the PRC1 complex is the one which is listed here. So here the core subunit. Now, when we trigger differentiation by removing the leaf, allow the differentiation of embryonic stem cells into embryo bodies, which recapitulate the early stage of animal development, since actually contain the three germ layer, the ectoderm, the mesoderm, and the endoderm. You can see the composition of PRC1 switch for this one, for example, containing CBX7, to in the EB, again, upon differentiation, which CBX7 is replaced by CBX2 and CBX4. And for example, if we force in more differentiated cells the overexpression of CBX7, what we observe is that these cells not only are incapable to undergo differentiation if we start overexpression from here, but if we force the expression of CBX7 at this stage, the cells actually can retro-differentiate and can gain some of the pluripotency characteristics. And this actually was recently nicely illustrated by the work of Gerald Dehaan together with Christian Helling, where they found that CBX7 overexpression in hematopoietic stem cells, which is an adult stem cell, the overexpression of this leads to enhanced self-renewal, so which means aberrant proliferation, and this would eventually lead to leukemia. So overexpression of the wrong subunit in the wrong cellular context would lead to human disease. Now, if we focus now on the PRC2 complex, what I told you before, those are the core subunit. And the question how the PRC2 is targeted is still quite debated in the field. We know that the RNA can play a role in this, and the role of the RNA can be either to prevent engagement of PRC2 to chromatin by binding to RNA. PRC2 is displaced from chromatin, but also other example, like for example, the role of exist actually can, or other non-coding RNA, they can favor the binding of PRC2 to chromatin. So RNA can indeed targeting or prevent targeting of PRC2 to complex, to chromatin. And we know that addition, so actually interaction of PRC2 with other Polycomb subunit, like for example, PCL, can facilitate the binding to chromatin template. And this is the case because, and this is actually due to the fact that the PCL protein are capable not only to recognize some histone modification, as I will mention in a second, but also they're capable of interacting with a stretch of methylated CPGs. So as you can see, Polycomb can be, the occupancy of Polycomb can be regulated by RNA, can be regulated by interaction of PCL protein, but PCL protein are not the only one which can interact with core subunit. For example, if PCL is replaced by JARI2, a different variation of PRC2 complex is formed. Remember, at the beginning I mentioned there is the PRC2.1 and PRC2.2. You can see here that actually, depending on the variation, we have specific subunit. And interestingly, the PRC2.2 variation, which contain JARI2, also is capable of recognizing HTM ubiquitination. As I mentioned at the beginning, this is a modification which can be catalyzed by the PRC1 complex. And again, here we go from PRC1 complex, depositing HTM ubiquitination, which is then recognized by the JARI2 within the PRC2.2 complex. So basically, with respect to PRC2, the situation is a bit simpler. We don't have so many variations like in the case of the PRC1. We have mainly two big variations. The PRC2.1, which contains PCL protein or Poly1, and the PRC2.2, which contains JARI2 and ABPP. And you will see in a second different biochemical characteristic. But can we prove really that these two complexes are mutually exclusive? Yes, we can. What we did, we performed a few years ago an experiment when we perform an EP for EPOP. EPOP is one of these additional factors for the core subunit. And we perform an EP for EPOP, what we found before all core subunits, which are shared between the two variations, the PRC2.1 and the PRC2.2. But there was a zero of the other protein, which also was found to interact with the PRC2 complex. So which means if EPOP is present, as you can see here, JARI2 cannot enter the PRC2 complex, right? And vice versa, if you do an EP for JARI2 complex, you see again the core subunit, but not EPOPs, which means they're two biochemically exclusive complex. Mutually exclusive complex. Now, how this is achieved under, if you look at the structurally speaking. So if this is the structure of PRC2 complex, which have been recently solved. And here you can see the ACH2, which is the set containing enzyme, which is the one which deposits the modification, the trimethyl group on lysine 27, and we have additional core subunit listed here, EED, SUS12, and RBP4. Now, this is a bit cartoonish kind of style, but again, the same subunit are listed here. And we know that there are many subunits that can interact with the PRC2 complex, but how this interaction is regulated. So we know the JARI2 interact with this region of SUS12. And once the JARI2 interact with SUS12, then it's capable of stabilizing the interaction of ABP2 to another region of SUS12, and this leads to the formation of PRC2.2 complex here. Now, if we look at other protein which I mentioned before, one is EPOP. EPOP also interact with the SUS12 and get exactly with the same region, the JARI2, with a very similar, partially overlapping region. And once EPOP binds to PRC2, also capable of bringing elongin BC, which I don't have time to talk about that today, and together with PCL protein, which also compete with ABP2 for the same regions. So now you can see from a molecular structural point of view that either one complex can be formed or the other complex can be formed because they fight for the same surface within the core subunit. Now, from the mechanistically point of view, we know that the core subunit ACH2 to the set domain leads to deposition of the trimethyl mark, as I mentioned before. And this modification in certain set of promoters, those promoters which are permanently repressed, also is share the presence of H2 monoubiquitination that, as I mentioned before, is recognized by JARI2. Well, vice versa, if we look at PCL protein, also I told you before that not only can recognize CpG island, unmethylated CpG island, but also active mark like K36 dimethylation and trimethylation. And these combinations of CpG island and active mark together with repressive mark, which are deposited by ACH2, it appears to be present at bivalent genes. And this is a set of genes which are genes which are important for cell development and differentiation. Now, another things that we recently noticed is that if you look at all human mutation, cancer mutation, they tend to clusterize in this region here. You can see this is a hotspot of mutation, and this makes sense because this is the region that contains the catalytic core of PRC2 complex. So mutation in the catalytic core, of course, would abrogate PRC2 function, or in other type of tumors will actually enhance PRC2 function, which both things can lead to misregulation. Another class of mutation is also present here. You can see here, and this is very interesting because actually these are mutations that might actually occur on this region where, remember, there is this overlapping interaction between either JARI2 or PCL protein, EPOP, and mutation in this region actually can favor one complex versus the other complex, and it can change, as I mentioned before, also the biochemical characteristic of PRC2. Now, basically, as I said, there are two main variations of PRC2, the PRC2.2 and the PRC2.1 with the core component here, and the specific factors are PCL protein here and JARI2 here. So more recently, we become interested in this family of protein called PCL protein. It's a family of protein because it contains 3 family members, PCL1, PCL2, and PCL3. So all of them contain a very interesting set of chromatin-related domain, Tudor domain, Ph.D. domain, and cellular homology domain, and chromo domain. Now, this is the new name, PCL1, PCL2, and PCL3. The former name are PHF1, MTF2, and PHF19. We got interested in particularly in PCL3, also called PHF19, because not only is overexpressed in, as you can see in a second, in different type of human cancers, but also because there is a short isoform, as you can see here, which lacks some of these very important domain, and actually the not having this domain prevent this protein to be inserted or participate within the PRC2 complex. Okay, so we wonder what happened if we now remove PCL3 right from the cells, if we perform an ablation of PCL3 from the cells. Would this lead to would the other 2 family members surrogate for the lack of the PCL3? So PCL1 and PCL2, would that supplement for the function of PCL3? Or maybe this whole complex become destabilized and the other complex become the major complex within the cells. So let me tell you a little bit what we have done in the past. So our lab identified PHF19 back in almost 10 years ago as an interactor PRC2 complex, is a protein which controls PRC2 occupancy, and also deletion of PHF19, which, as I mentioned before, is also called PCL3. Deletion of this protein alter the capability of embryonic stem cell to undergo differentiation. But more recently, what we found is that this protein, this PHF19, if we look in the database for cancer patient, we found that it can be either upregulated, right, like in the case of colorectal cancer, or downregulated, as you can see here, in patients with prostate cancer. And particularly, we put a bit more effort in characterizing the downregulation of PHF19 in prostate cancer model system. And this work was recently published in eLife, when we found that indeed the lesion, like when we simulate deletion of PHF19 in the proper context model system, we found that this actually lead to a reorganization of PRC2 variation, and I will come to that in a second, but also the cells, they change the characteristics. Deletion of PHfI9 in prostate cancer model system, what it happened leads to downregulation of proliferation, but the side effect of that is that the cells become more invasive and most likely also more metastatic. So these two things highlight the role of PHfI9 in mouse embryonic stem cells and some aspect of PHfI9 in cancer, but which is the role of PHfI9 in adult stem cells. So to address this question, what we did, we generated a mouse conditional knockout for PHfI9, and what we found that deletion of PHfI9 trigger the classical meiotic transformation. So many of Polycomb, deletion of Polycomb genes in the mouse models leads to alteration in the skeletal axis. So you can see here the vertebra C7, which in control animals and in humans, we never have ribs at this level. All the ribs start from the thoracic vertebra. Here, the deletion of PHfI9 leads to the formation of an extra ribs here, together with other malformation. So that was expected because, as I mentioned, other deletion of other Polycomb genes also have a similar effect on the skeletal axis. But what we did not expect is that the animals, when they become old, they develop splenomegaly. That was an indication that might have some alteration in the blood system. And when we look a bit more in details in the blood system, what we found is that PHfI9 knockout animal model, they have a lower cellularity, which means a lower concentration of cell per unit of blood. So these two things together suggest that there is a different, and most likely some issues, some problem with the blood tissue. And what we did, the gold standard technique for investigating alteration in the blood is by performing cellular transplantation. I'll guide you to this. So the initial point is irradiating the mice. By irradiating the mice, we completely kill all hematopoietic cells. So basically, the blood loses all its functionality, which means the animal will die unless this animal will receive a transplantation of bone marrow from a donor animals. So if we transplant bone marrow from the donor animals, which of course have not been irradiated, we can sustain the life of the irradiated animals. Now, what this also now allows us to perform a trick. Now we can compare wild type bone marrow versus bone marrow which have been isolated from animals which lack PHfI9, and asking the question whether bone marrow from animals which lack PHfI9 can actually sustain the survival of these animals equally good as the control bone marrow. So we performed this experiment, and we see no difference between control, which called flock here, and knockout animals. Then you can push the system and retransplant this one again from one side in order to push the system. And again, we don't see any difference. So basically, there is no significant difference between flocks and knockout animals. But when we perform the cellular transplant, third transplantation, then all animals which received the bone marrow from which lack PHfI9, all these animals were dying. So this indicated a defect occurs after the third transplantations, it means that the defect arises in the long-term hematopoietic stem cells. Let me put a bit in the context. If the defect would have arisen after the first transplantation, the defect will be most likely in more differentiated cells. In the second transplantation, would have indicated a defect in the precursor cells, in committed precursor. If the defect only arises in the third transplantation, which means the defect is here, the last set of cells which mobilized during this third round of transplantation. So the hematopoietic stem cells. So then we studied a bit more in detail hematopoietic stem cell, and what we did, we isolated hematopoietic stem cells using these markers indicated here, called the SLAM marker. And with these cells, which are very rare population of cells, what do we perform with the single cell transcriptomic as well as bulk transcriptomic? And the analysis indicate actually that if we look at the transcriptomic, there is a reduction in ribosomal biogenesis and the metabolic function. So comparing what the flux versus knockout PHfI9 hematopoietic stem cells. Also, lack of PHfI9 leads to a reduction in oxidative phosphorylation, a reduction in POL2 function. So these things together if you look in the literature, these things together suggest that actually lack of PHfI9 in hematopoietic stem cells indicate those cells are more quiescent. This is one aspect, and also what we noticed that there are some genes which are misregulated, which might indicate a kind of leukemia signature, but also we'll come back to this later at the end of my talk. So, but can we prove that the cells are more quiescent by using an orthogonal approach? Yes, we did. We performed Ki67 staining, and in knockout hematopoietic stem cells, the staining is reduced. And if we perform BRDU incorporation, these cells are capable of, and less capable of updating BRDU, which means that the cell are more quiescent. So this suggests that PHfI9 knockout leads to a more quiescent state if we look at hematopoietic stem cells. Yet, if we now, so that's what I meant. So there is a reduction number, which also both in LSK population, which is a bit more, it's not only hematopoietic stem cells, but also with some progenitors. But also, if we look at the very pure hematopoietic stem cells, you see a lower number of cells, which again indicate the cells are more quiescent. But then when we induce stress in these cells, and the stress can be done either by aging, or by transplantation, as I mentioned before, or by adding this drug, the 5-fluorouracil. In all cases, the cells which are more quiescent or in reducing number, they start proliferating, and they can proliferate equally well with respect to control, as you can see here, you can see here, you can see here. This is true for LSK compartment as well as for HSC compartment. In some cases, actually, the cells which are more quiescent, actually they compensate by over-proliferating. You see, the knockout cells can over-proliferate sometimes even better than control cells. So initially they're more quiescent. Upon stress, the cells can enter the cell cycle. But then the key question is whether these cells actually are able to undergo differentiation. So, so there's this question of what we did. We put in a single well, a single hematopoietic stem cell. So each well contains just one cell, and then we allow these cells to grow for we count for 4 days, every 6 hours. So in order to understand the kinetic of the growth, then after 9 days, we count the total number of cells, the colony size, right? And then after 14 days, we assess whether the cells were differentiating or not differentiating, or which lineage the cells were undergoing differentiation, right? So these three aspects, the kinetic of the proliferation, the colony size, and the cells under the type of cells we have at the end of 14 days. So I'm just here plotting the immunophenotypic characterization, and you can see here comparing the flux versus the knockout cells, there is a dramatic increase from almost 3% to 25% of cells which remain undifferentiated, which altogether indicate the cell are more quiescent. Upon stress, they start to undergo to differentiation, but then proliferation, but when they enter the cell cycle, they are not capable to reach equally efficiently the differentiation state. Most of the cells remain undifferentiated and remain as hematopoietic stem cells. So definitely there is a defect in differentiation. So this forces us to, I'm looking at the time I still have some time. So this forces us to go back into the, looking a bit more in detail in our transcriptomic analysis. And what we found is that actually between the genes which were upregulated, the pathway which were upregulated, one of the top one was actually the retinoic acid signaling pathway. Again, here you see RXR and RR receptors, as well as all signal pathway here. And you can see here, and that's what I mentioned before, knockout cells, the cells they remain more as hematopoietic stem cells, and the signature of stemness is actually overrepresented in knockout animals. The cells are more quiescent. Indeed, mRNA processing and ribosomal are downregulated in knockout. They are more represented in wild-type flux animals, and MIG target also downregulated. So all of these indeed indicate the cells are more quiescent. They are more stemness characteristic, and the retinoic acid pathway is upregulated. We perform validation, which I will spare you, and we validate genes of different categories, and all fit nicely. But also this recall to our mind the work which was recently published by Andreas Stram. What he found in his study, that actually the cells that we call the long-term hematopoietic stem cells can actually be subdivided into two types. In dormant long-term hematopoietic stem cells, inactivated long-term hematopoietic stem cells. And interestingly, in the work from Andreas Stram, the characteristic between dormant, or the difference between dormant stem cells and activated stem cells, are listed up here. So the dormant hematopoietic stem cells actually have a high retinoic acid pathway, activation of retinoic acid pathway, and lower biosynthesis and lower cymic activity in ROS production. And the activated one, which are this one, actually have exactly the opposite. The retinoic acid pathway is downregulated, and biosynthetic pathway and cymic become upregulated. So this somehow recall us actually the deletion of PhfI9 trigger exactly the same characteristic. As I told you, deletion of PhfI9 trigger retinoic acid upregulation, a downregulation of biosynthetic pathway, ribosomal activity, and cymic activity, and so on. So can PhfI9 actually the regulator between the dormant and activated state of hematopoietic stem cells? In other words, is PhfI9 controlling these two stages? And is that related to the variation between PRC2.1 and PRC2.2 complexes? So, and how this is achieved, right? Which is the mechanistic in this one. So what our hypothesis is that there are several PRC2 complexes, the PRC2 containing PhfI9 or PCL3, which control and binds to set specific genes, then the one containing PCL2 and the one containing GR2. Now, what we did in our model system, we delete this complex here. And we were expecting that those specific target would become upregulated, the repressive mark would disappear from these genes, right? Would be in a decrease of K27 trimethylation globally in the cells, right? And these genes would become activated. But paradoxically, what we found, instead of finding less K27 trimethylation, we found there was an increased level of K27 trimethylation. You can see this is an heat map, right? Clearly, by deleting PhfI9, instead of finding less K27 trimethylation all over the genome, we found increased K levels. But then we asked more specifically where this K27 trimethylation is accumulated. And it seems it's not accumulated at genes which control stemness characteristics. There is an equal amount, this is not significant. The difference is not significant. But then if you look at genes which are implicated in differentiation or transcription factor, which are implicated in hematopoietic differentiation, you see that it becomes significant. So it seems that this aberrant repression mark is specifically accumulated at genes which control differentiation. Here is some example. If you look at CBP alpha, PAX5, and EPO, all three genes are important for the several lineage of hematopoietic differentiation. Control cell, there is very little K27. Upon deletion of PhfI9, there is accumulation of K27, accumulation also of PAX5 and EPO. And this goes hand in hand with the fact that promoter become more compact as measured by attack, which is a technique which allow to measure accessibility to a given location. In this case, we are monitoring the promoter listen here. So basically what I've been telling you is that by deleting PhfI9, what we've done, we allow the formation of other Polycomb complex, right? The one containing PCL1, PCL2, even the one containing GR2. And those genes, instead of bivalent, when there are these formation of these new complex and these complexes are recruited to the wrong location of the genome, this lead to an accumulation, aberrant accumulation of K27 trimethylation, which does not occur everywhere in the genome, but is specifically for some region. In this case, as I show you, specific accumulated at gene set which control differentiation. And this leads to the hematopoietic stem cells to be more quiescent, but still they gain some stem cell characteristic. There is accumulation of K27, as I mentioned before. There is these genes which are important for differentiation. They are instead of being bivalent and ready to be activated, they become locked with this aberrant amount of K27 and cannot be activated. And this is true not only for the promoter region, but also in answer, which we look also in answer. Also, they accumulate K27 trimethylation. As I mentioned before, also there is a leukemic signature. And with respect to the leukemic signature, when we look either in the spleen or in the bone marrow, these control animals, you can see you have the myeloid cells population, B cells population here, T cells population here, while when we do the same analysis in PhfI9 knockout animals, here I'm showing you only the spleen, but as I said, the same is for the bone marrow, you see the appearance of this population here, which is not present here. So this is a new population, which is none of the classical one. It's not T cells, B cell or myeloid cells. It's a new population with a different characteristic. And when we assess the transcriptome of these cells, what we found that are very similar to a signature which is present in leukemic cells. And this is true if we use the signature from take from this publication or if we take from this publication. So it seems that it's the appearance of an aberrant population which have several characteristics of leukemic cells, or maybe most likely pre-leukemic cells. Sarantis, I wonder whether I still have 5 minutes. I'm running out of time.
No, you're okay. You can.
Okay.
If it was, yeah, you're fine.
Okay, I'll finish up with just a little follow-up story that we are developing. And so, as I said, the deletion of PhfI9 somehow predisposed the cells to a kind of pre-leukemic stage. And can this help us understanding what is a pre-leukemic stage? So we start to wonder, can we try to understand the pre-leukemic stage a bit more in detail? It's something that is not really well characterized. So for doing this, what we have done, and this now is in wild-type animals, and then we are doing the same in animals which are lack PhfI9. But to understand the pre-leukemic stage, we still need to perform this experiment in control animals, which are wild-type animals. And what we've done here, we have took two classical oncogenes. One is called PMRR, and the other one is MLL-F9. Both of them lead to leukemia, in human leukemia. These are more pediatric type of leukemia, this is adult leukemia, and this is a repressor, and this is a stronger activator. So two oncogenes with different characteristics, activator versus repressor, inducing pediatric versus adult leukemia. So we isolate the hematopoietic stem cells, and we infected these cells either with this oncogene or with that oncogene, right? And for simplicity now, I'm only show the data with respect to PMR alpha. And what we did, we allow these cells to grow over time. So from uninfected cells, mocked infected cells, and over time, we can follow the progression of this, the effect of these oncogenes in establishing leukemia phenotype in these cells. And at the same time, what we did, we take these cells, we inject in recipient animals, and we also see how this leukemia evolve in the proper context, so in animals in the presence of the niche. So those are the different stages: 0, 1, 2, and 3, and 4. The 4 is one in the in vivo. And what would we have done? We have done chromatin topology analysis, and what we see that in there is a dramatic reorganization of the chromatin, and this can be seen by a lot of active regions, which are actually large piece of the genome which are in the A compartment, the active compartment of our cells, this relocalized into the B compartment. Somehow you can say from the euchromatin to heterochromatin, if you want to simplify a little bit, while there is much lower movement from the B compartment, from the inactive compartment to the active compartment. It is actually in line with the fact we are expressing a stronger repressor. That's why large part of the genome from active become inactive. Now you can see some example here. So the active compartment is the one in yellow. This is the region of KL4, coisin, and MIR23, all genes which are very important for proper hematopoietic differentiation, which are in this mixed compartment, part inactive, part active. You can see the black and the yellow. But over time, from stage 0, 1, 2, and 3, 4, all these become repressed, all become moved into the B compartment. Now, this analysis was done in parallel, not only looking at chromatin topology, which you will see also more in detail later, but also we performed several omics quantification, which include ChIP-seq for some different marks like K27 acetylation or K27 trimethylation. We perform also DAC-seq, and I will provide you a few examples here. So now focusing again on KL4 genes, the one which I mentioned before, right, which become from A compartment, slowly switch to be a B compartment. the way in which you see this analysis, this is a 0 minus 1, and then 1 minus 2. It's a kind of progressive. You can see over time, there is a loss of K2. See, if it is below this line here, which means there is a loss of K27 acetylation. And this is true not only between 0 to 1, but also between 1 to 2. So it's a progressive loss constantly of acetylation. And this goes hand in hand with a gain of trimethylation, which means this region of KL4, which is an important gene for hematopoietic differentiation, lose active marks, again repressive mark, like the Polycomb mark. And this upon expression of the oncogenes. Now, if we look at the transcriptome, the global transcriptome, you see this the pseudo-time evolution. The cells from normal become slowly more and more altered until the full leukemic cells here. And again, the genes become upregulated, downregulated. There are many more genes which become downregulated, upregulated for the reason I mentioned before, since we are expressing an oncogenes. Now, here there are a few examples. KL4, the one I told you before, is expressed in stage 0 in hematopoietic stem cells, and then upon expression of the oncogenes, the expression goes down, progressively goes down, and the same is true for other genes, which I'm not going to list here. So some genes become downregulated, and some genes become upregulated. You can see here. GATA2, GATA1, both going up. c-KIT, which are marker of undifferentiated cells, also become upregulated. So we are losing differentiation, we are gaining the differentiation of these cells. Now, if we zoom in a little bit more in the KL4 region, you can see here, progressively, you can see there are two, I don't know how many of you are familiar with this, but there are two TADs, right? One clearly here and another one clearly here. And over time, this clear separation between these two TADs become a bit blurry. So there is an increased number of interaction, which are the one in brown indicated here between this TAD and this TAD. And simultaneously, you see the blue dots here. These indicate a loss of interaction between KL4 genes, which is the one, sorry, if we look here, this is a zoom of this region here. So this is the location of the KL4 coding sequence. And there is a progressive loss of interaction with region downstream. You can see one is here, another one here, another one here. And there is, because all these are blue, and there is an increased number of interaction within the locus itself. Now, if we compare this topology analysis together, we combine this one with this some modification, you can see that over time, there is a loss of interaction between this region. Sorry, this is a virtual 4C. There's a loss of interaction, you can see here, between the center of KL4 with this region here. In control cell are the one in red, and in black are upon expression of the oncogenes. There is a loss of interaction. You see the signal from here become reduced, and the same is true for here and here. And these three regions here, which I'm highlighting with this pink color, are actually enhancers, which progressively they lose the monomethyl mark, they lose acetylation mark, and they gain K27 trimethylation mark, which means that upon expression of the oncogenes, these lead to the compaction of K4 locus and the loss of interaction with the enhancer. And all of this leads to a repression of K4 expression. Now, the question is how much this also occur in human samples. If we see in the healthy bone marrow, K4 is expressed, but then if we look at patient with 15-17 translocation, which is PMR alpha translocation, KL4 is dramatically downregulated. While in other type of leukemia, this is not the case. So it seems to be specifically downregulated by upon expression of this oncogene. So now, obviously, what happens if we re-express KL4? Can this be used as a therapeutical approach? So what we've done, upon expression of PMR alpha, what we have done, we also, you can see here this over expression of PMR alpha, but also we force PMR alpha expression together with KL4 overexpression in a vector that cannot be silenced by PMR alpha. As you can see here, there is an increase of differentiation compared to the control with PMR alpha. There are almost no differentiated cells. If you look at CT11B positive cells, upon PMR alpha expression, there are almost no differentiated cells, but if we force KL4 expression, the cells are capable of regaining differentiation, and actually they stop proliferating. So PMR alpha leads to immortalization and progressive growth. If we co-express KL4, this growth is completely abrogated, so there is no signal here. So I'll finish here, and I just want to mention that work was done by here at CRG in Barcelona, and several person in the lab have contributed to work, are the one highlighted in yellow here. And also I would like to thank our collaborator that have been key instrumental for performing the experiment I've been presenting. And I finish here and happy to take some questions.
Thank you very much, Luciano. Great talk. I will start with a question from Marie Lamarck. How is the recruitment model for Polycomb complexes? Is it tissue specific or cell specific or both? And can both occur within the same cell depending on environmental cues?
So the question is
how is the recruitment? How Polycomb is recruited? How Polycomb is recruited? Is a cell or tissue specific?
Yeah, this is what is cellular specific is the composition of Polycomb complex. So depending on the cell type, you might have PCL1 or PCL2, you might have PCL2 or PCL3, you can have more of the GRE2 complex and so on. So these two complexes have different biochemical characteristics, that's for sure, but also they're different manner of being targeted to chromatin. And indeed, PCL protein are capable of interacting with K36 marks, so maybe target the Polycomb genes which are low expressed or middle expressed, but also interacting with CpG island, right? In the case of the variation of the one containing GRE2, GRE2 can read H3 monoubiquitination, which might go to a different place of the genome. Yes, it is cell specific, rather than I would say more than tissue specific, it's more cell specific, and that much depend on the expression of this additional cofactor of PRC2 complex. Now, how this is regulated is a question that no one has actually addressed so far.
Thank you very much. Gianluca Sigismondo asked if there is a different localization for GRE2 and EPOP. If you have seen a different binding and different genes that are regulated, I'm guessing by ChIP-seq or RNA expression.
Yeah, that's the problem of the ChIP-seq. Let's say,
or cut and tag.
Yeah, cut and tag or single cell ChIP-seq would have addressed this, because in a population there are many variations. So what we are planning to do is to perform sequential ChIP, right? Because in sequential ChIP you can only pull out whether they are both concurrently bound to the same place of the genome. That's something that at the moment is the only way to assess whether they are mutually exclusive on the same genomic loci, unless active Motif come up with a kit for doing single cell ChIP-seq. That
soon we'll be there. Thank you. Melanie, I ask you actually, how, when you do a PhfI9 knockout, you have seen, you get some cells that they are differentiated, but very few of them. Can you comment if it's a defect or a block in the molecular level, and how the cells overcome the locked repressed state?
Yeah, this also is interesting. So what we know is that, of course, there is a variation in the population. So not all the cells are the same. This is the stochastic variation, which is intrinsic in all cellular models. Now, in my opinion, there are the cells which lack PhfI9 are more refractory to undergo differentiation for the reason I mentioned today. So because the genes which are implicated for hematopoietic differentiation are somehow locked in this more repressive state. They accumulate more repressive part. So, and some of these genes can overcome this. And why is that? Because maybe they accumulate less mark, or maybe because when the signal for differentiation arrives, the cells is dividing, and at that moment the chromatin is more open and more accessible, and that transcription factor which might be present can actually access EPO genes or CBP alpha genes and allow transcription of these genes. It can trigger a cascade of events which eventually lead to full differentiation. But by and large, the majority of cells are more refractory to undergo differentiation. That is indication by from 2% to 24% of cells which remain undifferentiated, remain as stemness cells.
Thank you, Luciano. There are a lot of questions, and again, I apologize for